Hello students, welcome to my class. So this is the second video on the topic metal complex as drugs and therapeutic agents. So firstly we will study how gold is used for treating arthritis. Now you studied that different metal complex like platinum used as anti-cancer agent, vanadium is used to treat diabetes. Same way gold drugs were also used to treat arthritis. Okay. And this therapy using gold drug to treat arthritis is called as cryotherapy, and these gold drugs were given through the muscle, that is intramuscular administration of these drugs were done, and some drugs, that is gold drugs, were orally consumed also by the patient. So you should know some of the examples, that is one is gold trisodium thiosulfate, that is known as cyanochrysine, and another is gold disodium thiomalate, that is known as myochrysine, etc., etc. So there are several drugs that are given in order to gold drugs in order to treat arthritis. There's one drug which can be given orally, that is alkyl phosphine gold complexes. Okay, these complexes they can be given to the patient orally in order to treat arthritis. So usually this gold in the oxidation state +1, it readily undergoes disproportionate to give oxidation state 0 and 3. But in here in case of this gold drugs, so that it does not undergo disproportionation reaction, it is stabilized by using the gold drugs. This AU1, that is AU with +1 oxidation state, it is stabilized so that it does not undergo disproportionation using some suitable soft ligands. So the exact mechanism how these gold drugs they act against arthritis is not known, but it is understood that these gold drugs which are once administered into the body, they will be concentrated in your lysosomes present in the cells. Okay, so they will get accumulated in the lysosomes, and lysosomes they also contain hydrolytic enzyme which is responsible for the arthritis, that is which is responsible for the joint damage in the arthritis. These enzymes are responsible. So what does this gold drug do which has gone into the lysosomes is that it inhibits these hydrolytic enzymes. It makes sure that these hydrolytic enzymes cannot function, that is inhibition of this enzyme, and that's how it helps to treat arthritis. And another way is that its action is also seen against infectious agent, that is it is also seen to act against the infectious agent that cause arthritis. So these are the two different ways in which they work. So these are some of the structures of the gold drugs, that is sanochrysine, myochrysine, solganol, etc. So you need to know at least three of them to draw the structure of some of the gold drugs. So what exactly do the gold drugs do? You know that they enter into the lysosomes and they act, but here they're telling us a little detailed mechanism of what these drugs do is that they go into your joint. So arthritis is nothing but pain in the joints, correct? Because of problem in the bones, there is pain in the joints. So what does this drug do is that it goes into that area where that joint pain is there. It goes into that joint structure and it stabilizes the collagen. So collagen is a type of protein that is present in our body, especially in our bones. Okay. So for bones and joints, bone strengthening and joint strengthening, collagen protein is required. So that is where it is present. So what does this drug do is that it directly goes towards this collagen site and it stabilizes the collagen by cross links. Okay, so also the evidence indicate that the gold drug it protects the SH groups of this protein and inhibits the formation of disulfide bond. If there is disulfide bond formation in the protein, what happens? That it results in denaturing and it results in the formation of macroglobulins. So if the protein, the collagen protein that is holding the bones together, that is helping to strengthen the bone, if it gets denatured and if there is formation of macroglobulin, it can lead to arthritis and pain in the joints, correct? So what is this gold drug doing? One is it is going there and it is stabilizing this collagen by cross linking, and it is also protecting the SH groups of the protein so that it is not substituted by some other metal, toxic metal or heavy metal, and it does not form disulfide bonds. So that is how this gold works. Another possible way to treat arthritis is that one more reason for arthritis is that sometimes this copper and zinc containing metalloenzyme might be inactivated because they are SH groups. It's a metal enzyme, correct? So it will have SH groups in it. So these enzymes containing copper and zinc, they might be inactivated because of their SH group binding with a poison, we can say, called as thionine, which is coming like a toxic agent into the body, and it is binding with the SH group of the metalloenzymes containing copper and zinc, because of which it cannot function properly, and because of their malfunctioning there is diseases like arthritis, etc. Okay, so what does this gold drug do is that now it has to treat that. No, so it will go and it will bind more tightly to this thionine, and so that it releases, it is released from the SH group of the metalloprotein, and the metalloprotein group and metalloenzyme is now free. Okay, so what is a gold drug doing? It is going and binding more tightly with the thionine so that it leaves the SH groups of the metalloenzyme, and the metalloenzyme is free to function. What happens if the metalloenzymes SH groups are attached with thionine? They become deactivated. You know that when enzymes SH groups are replaced by toxic metals, they become deactivated. So they have shown it in reaction form. So this is your active enzyme which was performing properly in the beginning. Now that thionine poison will come and it will attach with the SH group of the metalloenzyme. See, you can see here the H group here is substituted with the enzyme part, correct? So this entire enzyme has now become inactive. Okay. Next step, we are taking the gold drug in order to treat this inactivated enzyme and make it active again. So on treatment with the gold drug, what happens? This part, that is thionine part, is removed off and the enzyme is retained back. And you can see the thionine forms a complex with the gold drug, which is then removed out of the body. So there is a restoration of activity of copper and zinc containing metalloenzymes. So what are the two groups, two ways in which the gold drug is acting? One is the stabilization of the collagen protein, that is it will make sure that it does not form disulfide linkages, and one more is by activating the copper and zinc containing metalloenzymes. So apart from this, it was seen that gold drugs, they have effect on lymphocytes, monocytes, and neutrophils, and inhibition. They can also do inhibition of T cell proliferation and of polymorphonuclear monocyte activation is also done by this particular drug. Now, what is important is the next point. Okay, that is in vivo, that is in some organisms, the gold drugs were seen to form a complex with the cyanide, that is AuCN2 minus. Okay, and the CN that is required was obtained from SNCN minus by the action of peroxidase. Okay, so in organisms, the gold drug, it formed AuCN2 minus complex, that is from the CN was extracted from SCN minus. Okay, and in this was seen in some of the organism, whereas in case of humans, if a patient is a smoker, if he has the habit of smoking, then some CN minus content will be there in his body because of smoking. Okay, and so if that person who is smoking also has arthritis and has undergone gold metal complex treatment for arthritis, then what happens? That person's body already contains CN minus, and AU is also there in the person's body because he's undergoing treatment, correct? So there is a formation of this complex in that person's body. So such people who have the habit of smoking and also are undergoing treatment for arthritis, there is a high chance of formation of AuCN2 minus in their body, and they have to deal with the side effects of this formed complex. Now, this complex that is formed, that is gold with cyanide, has lot of side effects. So such a patient might suffer from these side effects. That is this AuCN2 minus that is formed. What does it do? It attacks the immune cells involved in the inflammatory process. So that and also it can enter the cell and limit the oxidative burst. So there are certain side effects that is formed from this complex that is formed by gold and cyanide. Okay. And one more thing is that in the body, the gold mainly exists in +1 oxidation state. Okay. So it was seen that even if gold was administered in the +3 oxidation state, the amino acid that having SH group reduced this gold having +3 oxidation state to +1 oxidation state. Even the disulfides that is present in our body, they reduce this Au3 to Au1. So usually in the body, the gold always existed as Au1. Next topic is how these metal complexes, they are used as radio diagnostic agents. Now students, here we are studying how different radioactive isotopes of different elements, they are used, one in the diagnosis of diseases and two in the treatment. So several radioisotopes, they are used in radiotherapy as well as radiodiagnosis. Radiotherapy means treatment and radiodiagnosis means finding out where what part of the body has the tumor. That is called as diagnosis, and what kind of tumor it is, how big it is, and then it is made to undergo radiotherapy. So the radiotherapy, what do they do is that they take this radiation, mainly the gamma radiation that is used to destroy these diseased tissues. Okay, so gamma radiation can comes out from many of the radioisotopes. So one of the radioisotopes of cobalt, that is cobalt 60, it can destroy tumor cells. What are tumor cells? They have their DNA is mutated, mutation of DNA takes place, so they are, those type of cells are tumor cells. So on subjection of gamma radiation, this DNA will be completely destroyed, so it will lead to the destruction of the tumor cells. Also gallium has a very wide application in this radiotherapy. That is, gallium citrate is administered into the body, and what does it do is that it goes and accumulates near the tumor, and then that's how we will be able to detect where the tumor is. So gallium is not just gallium 67, it is not just used for detection of tumor, but it also destroys the tumor cell. So how does it happen is that gallium citrate is made to administered into the body. Okay, it is sent into the patient's body having tumor, and in the body it is converted into gallium transferring complex. Okay, and these transferrin, the gallium transferrin receptor sites, they get accumulated, they try to accumulate the gallium 67 in the tumor cells. Okay, so what happens in the tumor cells? There is accumulation of gallium 67. What is happening? Gallium citrate is sent into the body. In the body it is converted into gallium transferrin complex, and the transferrin receptor sites, what do? They accumulate this gallium 67 in the tumor cells of the body, and that is how this gallium 67 by radiotherapy can detect the tumor, how big the tumor is, where exactly is the tumor, and also it can destroy the tumor cells. Also gallium 68 complex, it can be used for brain imaging agent, and any tumor in the brain can be treated by gallium 68. And lipophilic gallium 68 complex, they can be they are accumulated in the heart, and they can be used for this heart scanning, that is PET scanning. So these are some of the applications. Other two major isotopes that are required in the detection of tumor in the brain. This is one is iodine isotope, that is iodine 131. One more is phosphorus isotope. Okay, so these two isotopes, they are used in detecting and treating the tumor in brain. Okay, so the gamma rays that is coming from iodine isotope, it can pass through the skull of your brain, and the location, one location of the tumor can be detected approximately. So what happens here is that first the iodine gamma rays is made to pass through the cell, and it will give you one particular area, area in which that tumor might be present. It does not give you the exact location of the tumor, but it gives you one area, so much, half this part of the brain, left half of the brain, or back side, or like that, where that tumor may be present. Then what they're doing during surgery, they're doing using this phosphorus isotope. This phosphorus isotope will have short range. Okay, the beta rays coming from this isotope, they have short range, because of which, because of it having a short range, you can detect the exact position where that tumor is present. So first, using the iodine isophore, you can approximately tell where it is present. Then during the surgery, in that region where the iodine isomer has shown you that tumor place, you are using this phosphorus isotope and checking where exactly in that position the tumor is present, and then the tumor is being removed. Next, so another example for a radioisotope is TC99M, that is technetium containing radioisotope, which is required for, cerebral blood flow imaging, renal function imaging, cancer imaging, and myocardial perfusion imaging. So different types of imaging of body or affected organs of the body can be done by this technetium containing radioisotope, and it can get accumulated in the heart, so you can get the heart images, PET images of the heart. It can also get accumulated in your bones. Okay, so bone imaging can also be done using this isotope. Other than that, this isotope is also used along with the anti-tumor antibiotic. What is an anti-tumor antibiotic? Any antibiotic that is sent into the body in order to treat the tumor that is present in the body. So this anti-tumor antibiotic its property, or its activity is enhanced when it is present with this radioactive metal. So it is incorporated into this anti-tumor antibiotic and then sent into the body. One example they have given is bleomycin. Along with bleomycin, this technetium radioisotope is incorporated and then sent into the body, because of which its anti-tumor activity gets enhanced. So, and this is a diagram of the technetium containing radioisotope, which is used for treatment of heart, which is used for heart imaging agent, the tumor which is used for detection of tumor present in the heart. So this technetium is very widely used in the medical field in order to image, for imaging purpose. What is imaging? Why is it imaging so important, students? Because any tumor, any doctor, first you go, he has to detect where exactly is the problem present. Correct. For that, imaging of that tumor is very important. So technetium is one that widely used radioisotope, which is used for the imaging purpose. Same way, if you have some bone crack somewhere, what will they do? They will take an x-ray and they can detect the exact position where that crack is present in the bone. Correct. Same way, in order to detect where the tumor is present, imaging is very important, which is done using this technetium isotope. Now, where is this technetium isotope obtained? Is from MO42. When it undergoes beta decay, you can get technetium isotope. So this technetium, separation of this technetium from this MO42, it is done on the basis of cow and milk system. Okay, so the parent nucleus here is this MO99. Okay, and technetium, when MO99 it undergoes beta decay, it will give you technetium 43. So MO90 here, we said you can discuss cow and milk system, how MO99 gives out technetium can be understood by using cow and milk system. So here they're telling that MO99 it acts as a cow, and this can be considered as a milk. So this is just an example they're giving for you to understand. So here MO99 is acting like a cow, and technetium 99 is can be considered as its milk, and it can be milked for every once in every 24 hours. That's how you can get technetium 99. Same way, isotopes of sodium, they can be used to detect the site of obstructions of blood circulation. So there is any blockage in the blood circulation, then that can be detected by sodium containing radioisotope. And iodine radioisotope can be used to diagnosis and treatment of thyroid. So here in this topic, you need to know what type of isotope is required in what type of application, what curing what type of disease or what type of diagnosis. Then one thing is that the requirement here for the radioisotope that is used in diagnosis is that they should be short lived with low energy gamma photons. What will happen if they are not short lived, students? They will retain the body, because we are sending these radioisotopes in order into our body, correct? And you know that radioisotopes have both good and bad side to it. So, accumulation of radioisotopes in the body can actually result in the person's, it can be like a adverse effect to the body. So, instead of treatment, it can work in the opposite way. That is, the person might get affected further. So, these radioisotopes, using them in treatment should be very, very carefully used, and they should have low energy photons, that is low energy radiation, which should not cause further mutation of the DNA present in the body. So, what are the requirements for a radio diagnostic agent or radio isotope to be used as a radio diagnostic agent? This is very, very important. That is, they should be, first thing is they should be short lived. That is, their lifespan should be very short, so that once they are gone into the body and the tumor is detected or destroyed, then they are also destroyed as well. And they should radiate only low energy gamma photons, otherwise they can damage the normal cells of the body. So they should not damage the normal tissues which are already in proper condition. They should only destroy the tumor affected tissues. So if they give out alpha or beta particle, they will destroy the normal tissues. So they should only radiate low energy gamma particles. And the degraded product, that is, once they give out these radiation, they become degraded. So these products that are degraded, they should be excreted. They should not be toxic to the body, and they should be easily excreted from the body through urine or some other forms. And they should also be available in suitable forms, so that they can be administered into the body of the patient, and they should be stable enough so that they can enter into the body and they can reach the required target site and do their work. So these are some of the requirements of a radioisotope to be used for as a radio diagnostic agent. If it does not satisfy all these requirements, then it cannot be used as a radio diagnostic agent. Next, one more important example is that of boron 10, which is used in neutron capture therapy. It is used in the treatment, diagnosis, and treatment of tumor, especially in the brain tumor. Okay, so what is neutron capture therapy, students? Here, boron 10, it can capture the slow neutrons. So, whenever there is this radioisotope boron 10 present, and if you signal or if you give out low capturing if you give out slow moving neutrons, this boron 10 radioisotope can capture it, and by capturing it, it can release short range alpha particles. So here they've given that example. This is a slow moving neutrons, boron, radioactive boron isotope. It captures this neutron and gives out alpha particles. Okay, and this alpha particle that is given out, it can be used to destroy the tumor cells or the cancer cells present in the body. Now you already studied that the radioisotope should only give gamma radiations and not alpha and beta, because alpha and beta can affect the body. But only in this case, that is in case of boron, the alpha particle, they will not destroy the normal cells. Okay, only because they are of short range order. These are short range alpha particles that are given by boron on neutron capture. So they will not destroy your normal cells. They will only destroy the cancer affected cell. So how do they do this is that first the boron radioisotope is administered into the body. It will go into the body and get accumulated in the tumor cells. Okay, then from outside there is slow neutrons that are given out from the outside external source. Okay, so these boron, when that slow neutrons are given out from the outside, what do? They try to capture those neutrons, and as soon as they capture the neutron, they will give out alpha particle or alpha radiation. These alpha radiations, when they fall on the tumor affected cells, they will get treated. Okay, so based on this principle, what do? Attempts are made to incorporate this boron containing isotope in suitable antibiotics, which is then sent into the body to get attached with the neoplastic cells. This is how boron is being used in the treatment as a radioisotope. Next, we are studying about antimicrobial activity of metal chelates. Now, we already studied what are chelates or chelating ligands and how they detoxify the body from these poisonous metal. But here we are studying how are the antimicrobial activities of this metal containing chelates. Okay, so one thing you have to remember is that these chelates or chelating ligands, they cannot function if they are not having a metal ion. So the metal free chelating drugs administered require the complexation with biologically available metal ion to show their activity. So without a metal ion, they will not function. They require a complexation with the metal ion in order to show their antimicrobial activity. So we are studying about different antibiotics containing metal chelants and how they show antibiotic activity. First is the ionophore antibiotic. Now ionophore you already studied, they are used in transport of metal ions across the membrane. So this can also be used for antimicrobial purpose because of their metal binding properties. So these ligands ionophore, what do? They wrap the metal ions. So these are not found very useful in case of mammals, except for in chickens. It is used to treat coccidiosis, but majorly in mammals it is not of much use. Why is this happening is that these ionophores, they can change the membrane permeability, and you know that as soon as the membrane permeability changes, the membrane allows accumulation of excess of ions into the membrane, or lot of ions can enter the membrane, water can enter the membrane. So because of the permeability of the membrane changing, the property of the cell changes. That is, cell can get swelled up because of excess water or other ions entering into the cell. So, ionophore antibiotics are not widely preferred in case of mammals, because in case of mammals they can show adverse effect. One example they have given is valinomycin. It is highly, when it is the cell membrane, whenever it has valinomycin, it becomes more susceptible to K plus. That is, valinomycin has high affinity for K plus. You know that, correct? So if valinomycin is attached to the cell membrane, that cell will have more susceptibility towards K plus. So the cell's permeability will change, and excess of K plus will come into the cell and get accumulated, which can damage the cell. So ionophores are not very popularly used in case of mammals. That is a reason. Next is antibiotics like tetracycline, streptomycin, etc. They are known to have potential metal binding property. That is, they will have metal binding sites. Okay. So how do they work as antibiotics in the body? Now, students, these and transfer in antibiotics, that is this tetracycline or streptomycin, they work on the principle, they interfere with the Fe metabolism of the bacteria, because these metal binding property is utilized by these antibiotics to show their biological activity, and these metal binding sites, they are delicately balanced so that they can successfully compete with the metal binding sites of the bacteria while not disturbing the host. So now what is the bacteria? Let us understand what they are trying to tell in this. What will the bacteria do, students? Bacteria will come, and Fe is present in your hemoglobin, myoglobin, and different proteins. Correct. What will the bacteria do? It will go and attack that central Fe metal, and because of which the hemoglobin, myoglobin, etc. might get deactivated. So this tetracycline, it is an antimicrobial drug. What does it have? It also has high affinity for metal ion, and it has delicate metal binding sites which are delicately balanced. So what will it do? Before this bacteria goes and binds with the Fe, it will compete with that bacteria, and before that bacteria binds with the Fe, it will go and bind with the Fe, so that bacteria cannot come. Once this is bound to the Fe, the bacteria cannot come and bind and deactivate that Fe containing enzyme or protein. Okay, that is how they work. So you understood, they are competing with the bacteria because they also have good metal binding sites, so they are competing with the bacteria, and before the bacteria binds to the target site and inactivates it, they will go and bind, and that's how they will protect that particular enzyme or protein. So here they are explaining how exactly this tetracycline works. So its antibiotic activity of this tetracycline drug, it increases with the it increase in the metal binding ability. You know why? Because previously we said that its metal binding ability, because of that ability, it goes and binds, it competes with the bacteria and binds to the metal before the bacteria can come and bind. Okay, and it has hard donor oocytes, okay, as chelates, which will prefer hard cations like calcium and magnesium. So one side effect of consuming this drug tetracycline is that the person will lose his will affect the coagulation of blood. What is coagulation of blood? If you start bleeding somewhere, your body, your blood will have coagulative property. That is, after some time you can see that the bleeding has stopped and the blood coagulation has taken place. That is, blood has formed a protein like thing around that tone or around that cut, because of which the blood has stopped. So this prevents excess of blood loss from the body, and this coagulation is done because of calcium that is present in the blood, okay, that is required for blood clotting. But tetracycline, because it has hard donor oocytes which prefers to bind to hard cations like calcium and magnesium, what does tetracycline do? When it comes into your bloodstream, it can take the calcium from your blood and form complex with calcium. So the person will not, because his blood is deficient of calcium, he will not have blood clotting property. Okay, but majorly why is this tetracycline still used is that the action of tetracycline is directed towards a ribosome of the bacterial cell. Now imagine your bacteria already has your blood, it already has bacteria in it. So what is a you have taken tetracyclic drug in order to kill this bacteria that is present in the blood. So what does tetracycline do is that in bacteria, for their ribosome to get stabilized and for their ribosome to function, they require Mg2 plus. And now you know that tetracycline has high affinity towards Mg2 plus. So when it comes into the blood, it removes this Mg2 plus from the bacteria's body and forms a complex with this Mg2 plus. Because that Mg2 plus is removed from the bacteria, the ribosomes of the bacteria cannot stabilize and cannot function. Therefore, eventually the bacteria will die. So they conducted an experiment and they saw that Mg2 plus binding hypothesis is correct, and it is evident from the inhibition of growth of E. coli by tetracycline. That is, when they tried to grow E. coli in a petri dish, and when they put this tetracycline drug, it was seen that E. coli could not grow. That is, there was inhibition of growth of E. coli. Why did this happen? Because Mg2 plus from the E. coli was removed by tetracycline and it formed complex with that Mg2 plus. So this is how tetracycline will affect the ribosome of the bacteria, and that's how it will kill the bacteria in our body. That's why it is used as a antimicrobial drug. Next is auxines. These auxines and substituted auxines, they show antifungal and antibacterial activity, especially when the auxines, they have lipophilic group. You know that because of lipophilic group, these drugs, they can enter into the cell membranes and they can penetrate into the brain, etc. That's why because of lipophilic group, they become, their activity, that is antimicrobial activity, will be enhanced because they can penetrate into the cell membrane because of the presence of lipophilic group. And for the auxines to function, what is very important is these two ions, that is iron and copper is essential. So this complex in 1:1, if it is present, then auxine will function. And what do? They catalyze the oxidation of cellular mercapto compounds by cellular oxygen. So if you know this much, it is enough why auxines are used as antimicrobial agents. Next, it was seen that some of the microorganisms like Staphylococcus, etc., they have become very much resistant to the antibiotics. That is, the antibiotics that you're consuming in the body in order to get rid of these bacteria or microorganisms are not working enough. So since they have become resistant to it, so you can use chelation therapy. So some chelates, new chelates that are introduced like iron containing chelates or ruthenium containing chelates, they can be used. They are seen very effective towards these bacteria. Okay. So how do they work? They work in two different mechanisms. So what do is that these cationic complexes, that is these chelates, they occupy the sites which are normally utilized in the initiation of the infection on the host cell. So there will be a site in the host cell where the infection will be initiated. So what will these chelates do? They will go and bind in that site so that the initiation of infection itself will not takes place. That's how infection can be stopped. One more way is that the first step, that is, if the infection is already initiated, what happens in the first step is the absorption reaction involving electrostatic interaction. So these acetylcholine, this is a type of bacteria, it has positively charged head, because of which it interacts. There is electrostatic interaction seen between this acetylcholine and the host cells. Okay, but these cationic chelates, they also have positive charge on them. So what do? They may block the acetylcholine receptor through competitive inhibition. That is, before this positively charged head of the acetylcholine interacts with the host cells, in competition to them, this cationic chelates will go and they will go and interact with the host cells, because of which these acetylcholines can no longer interact with this host cell. So neutral complex have no effects because acetylcholine has a positively charged head, so you need a positively charged chelate to do its work, because it should do the same work as that of the bacteria is doing, so that it can block the bacteria from doing it. Correct. So that is the second way in which this chelating drug it acts. Next is isoniazides. These are drugs that are used to treat tuberculosis. So they are anti-tuberculosis drugs. And it was seen that the chelate, when it forms complex with the metal, that is when chelate is present along with the metal, the drug is much more effective than just the free ligand or free chelate. Okay, so the activity is enhanced. What is the reason, students? We have studied previously also. Whenever a metal is bound to the chelating complex, that drug is very active. Reason is because it becomes lipid soluble. Therefore, its complexation property increases, and this drug can penetrate through the cell membrane, so its activity is enhanced. That's what they're telling. So one drug that is used in anti-tuberculosis, that is thiacetazone. Its property increases when it is forming complex with Cu2 plus, so metal complex that is Cu2 plus forming with this drug thiacetazone makes it a better tuberculosis drug. And also some other examples they have given, that is one is kojic acid and bacitracins and sodium methyl dicarbamate. All these drugs are used to treat tuberculosis, but their activity becomes enhanced through metal complexation. That is, if metal is incorporated into them, they become their activity becomes enhanced. So there's a this is the representation of kojic acid, and this is oxines. Next is bleomycin. This is a glycopeptide drug. It has antiviral, antibacterial, and anti-cancer properties. So you know that cancer means there is mutation of the DNA. So what does this drug do is that it will cleave the DNA helix of the cancer cell. That is, it will repair this DNA which has undergone damage because of cancer. And but for it to actively do this, for its activity to take place, it should have iron metal in it, without which it will not work. If iron is not there, this drug cannot work. And some other metal ions like cobalt, copper, zinc, they reduce the activity of the drug. It is seen that these metal ions, what do? They compete with the iron in order to take the place of iron in this drug. So iron will be already incorporated in this drug, that is bleomycin, whereas this cobalt, copper and zinc, they compete with this iron to take its position. Okay, and when they are substituted, when it is substituted by some other metal apart from iron, it cannot function that well. And this iron bleomycin complex, it reacts with the cellular oxygen to generate the cytotoxic agents like hydroxide radicals and superoxides. These are responsible for some of our biological activities. So it was seen that the reaction of this O2 with the Fe BLM complex, it gives another complex, that is BLM FeO. This species is generated, and this is responsible for DNA destruction. That is how the DNA that has got mutated because of cancer can be repaired. That mutated DNA can be destructed. And other examples are the anti-cancer activity of thiosemicarbazones. It occurs when it is in complex form with the copper metal, and other like antimony, arsenic, and mercury compounds. They also, although they are considered as poisonous elements in our body and should not be taken because they are heavy metals, still they can show some antimicrobial activity. How is that? They can bind with the SH groups of the microorganisms, and that's how they will inactivate the microorganisms or pathogenic microorganisms which from infecting the body. That's how they work. This is the exact same way how these microorganisms work in our body to inactivate the enzyme. Same way what these heavy metals are doing, they will replace the H group in the SH groups of these microorganisms, and they will bind there, and hence they will inactivate these microorganisms from attacking or continuing their activity in our body. So although these are toxic, but in a very very small minute amount, they can act as antimicrobial agents. Next, the last topic is about an antidote. This was a topic that was left out in the other part. So, what exactly is an antidote and how is it used to convert poison to non-toxic product? So, antidote can be any substance that will work against the poison that you are taken. So, it is coming from a Greek word which means given against. Any poison that is consumed, it will act as counteract against this poison present in the body. Okay, so it limits the effect of this poison in the body. So there are three types of antidote. One is mechanical, second is chemical, and physiological antidote. In mechanical antidote, it prevents the absorption of poison into the body. Example is activated charcoal. By absorbing, it will prevent the absorption of poison into the body. It itself absorbs the poison on itself. That is activated charcoal. And some other examples are there. In case of chemical antidotes, they change the chemical nature of the poison and they prevent it from, they make it non-toxic in nature. For example, sodium thiosulfate, it changes the toxic cyanide to non-toxic thiocyanate. Last one is physiological antidote. They, what do? They counteract the effects of the poison by producing opposite pharmacological effects. So whatever effects were expected, they counteract it. That is, um, one effect, if a person is lacking something, that counter effect is done so that the person will not suffer because of poisoning. So they do the counteract of that. So one example is sodium nitrite. It converts hemoglobin into methanoglobin in order to bind to the cyanide. That is one example. Next is the mechanism. How does this antidote work? One, it works with complex formation. Second is metabolic conversion. So in complex formation, what happens, students? Here the antidote forms a complex with this toxic substance, and because it has formed that complex with this toxic substance, it is like it is trapping that poison so that it is not free to interact with the receptor or to enter the cell membrane. That is complex formation. That is antidote that you're giving into the body is forming complex with that poison, because of which the poison is not free to enter the cell or to interact with the cell. Second is detoxification, metabolic conversion. What conversion takes place? That poison is converted into a less toxic substance or less poisonous substance. This is seen in case of nitrites. They interact with the hemoglobin and cyanide to form cyanomethanoglobin. That is, hemoglobin that is infected or bound with cyanide, it's made, cyanide is made non-poisonous by the detoxification, that is conversion of cyanide to uh, methamoglobin. Okay, that's how cyanide poisoning can be prevented. These are the two mechanisms in which antidotes work. Next is how exactly is the pathway of antidote functioning. First is it forms a complex. As soon as it gets into the body, how it forms a complex with the poison. Then there is metabolic conversion, that is conversion of toxic to non-toxic. Then there is prevention of toxic metabolite formation. That is, from that toxic poison, there is no metabolite formation. That way, it will prevent the formation of toxic metabolite. Then it also changes the physiochemical nature of the toxin so that it will no longer be toxic in nature. Then antidote returns to the normal function by repairing the defect. So this is a mode how this antidote works, and flow chart is also given how the antidote works. Finally, the antidote is excreted from the body. So with this, we have completed with the paper, that is bioinorganic chemistry, and this is a very good topic in order to study and understand, because you're studying about your body and how about the biological system. Um, I will be ending this video here. Thank you for attending my class. Take care and bye.